let me say, because now it's about a participative
Yes.
project, right? And
Yes.
just say to remind you that this was done for the first time. And as you know, we always had we had an intention to have a system where we can, where the C3C can contribute to the PhD work. We know very well that there is a large majority of work that where there is no projects, and it's very difficult to have all the funds to do and to perform all the studies that during the PhD. And so we decided for this year to try what we call Projeto Participativo. And the main idea was that we open a small call, an internal call, just for PhD students where they applied, and a very simple application. And then what it was interesting, at least for me, this is not new, other research centers did, so I would just also use this idea, or part of the idea, was that the selection was not done by a panel, but it was done through election, so a vote from all the members, the integrated members of the research center. So this is that. This is why it's participative. So everyone could select the project that likes more, and somehow it was the favorite one. I hope for the good reasons, of course. I think that it's always, we manage not to have just one, but to have three. I think it's very important to have more than one. And I'm sure that next year we will, let's say, increase the quality of the system, because it was our first attempt. But this is something that we want to maintain, because I think it's very good to have this type of initiatives within the research center. And now, Gabriela.
Yes, thank you, Cristina. Yes, basically you already said what this is all about, and it's a very interesting initiative, I think. So it's very nice to have this. So then we invited these three awardees, let's say that, to talk about their PhDs work here for us in 10 minutes each one. And we are a little bit ahead of time, but I believe that the three of you are here, so I think we can proceed. And we are going to hear about your projects that have been a little bit financed by this participatory budgeting, like in the cities we do, right? So I invite the first speaker, Raquel Mendes.
Yes, I'm here. Can you hear me well?
Yes, very well. Yeah, nice. You can share your screen, please, and the floor is yours.
Sharing. I guess you can see it, right?
Yes, we can.
Okay. So thank you for having me here. I'm a PhD student integrated in the Conservation for Socio-Ecological Systems. I'm here to present this project called Biodiverse Schools, assess the connection of children to nature and promote local biodiversity. This is within my PhD project that is supervised by Professor Margarida Santos Reis and by Professor Martin Schoss from the University in Vienna. So just a brief introduction. We know that this connection, our connection, adults and children with nature, can provide us different benefits. But for children, these direct experiences in natural worlds are very important for different levels of their development, as you can see here on the right image. So we also know from the literature that the younger generations, for the last couple of 2 decades or 3 decades, they are becoming more disconnected from these natural worlds. Some authors even called this particular author, called Richard Louv, called this a nature deficit disorder, and him and other authors have highlighted that these have the different several negative impacts, and we can attribute this to the increase of the urbanized world, and also from the less time we spend outdoors. But there are other main causes. For instance, we have like the information we share with children, not only particularly in schools, but in the other environments as well. But we also have the disinformation associated, for example, with myths. In this aspect, we have the technology, particularly the excess of technology, because we know that technology can also have a positive and increase the participation of particularly the youth with nature. Also, the lack of opportunities, being the less time we have, adults have, or the less opportunities, for example, when children don't have access to natural or green spaces around the places they usually go or live. And for these last couple of years, we have another situation here, that is this pandemic situation that is not improving this disconnection of children with nature. So how can we improve this and reconnect children and nature? I will particularly focus here in the case of schools. We have seen, and other authors have seen, that green or greener schoolyards can also have multiple benefits. We see here in this left image that it can also, not only for children, but it can also be beneficial for the environmental and also for the communities that are surrounding the schools or families, for example. And we also know that these green spaces, these green schoolyards, are very important, but usually they are underused or non-existent. But they are very important for children, for their learning process and engagement in nature. Here in these right images, you see an example of a project called Green Schoolyards America, when from 2006, from 2019, they have this beneficial and this development in their schoolyards into a more natural environment, so that kids can also enjoy and learn in the outdoors. Here, there is another example that I've used for my application. It's a project called Space to Grow. And you can see here that the schoolyards, the outdoor environment in schools, they can be used for different aspects, not only for children to play, for sports, but it can also be used, for example, the vegetable gardens can be used for opportunities for children, not always to learn about how to harvest healthy food or foods, but also to learn about the process, to learn about the biodiversity that lives in these spaces, and also for other aspects to increase the environment and benefit the environment, and also as well the biodiversity, the local biodiversity in these spaces. So within my PhD project, that it's called Reconnecting Children with Nature Using Biodiversity and Education for Sustainability in Lisbon Schools, the main goals are to understand and evaluate the social factors that promote nature connection and conservation awareness in children. And I will be using the biodiversity and environmental programs, not only in schoolyards, but also in further in the other nearby parks and also in the Monsanto green area. It has these 4 main tasks. It starts with the textbook content analysis that I'm currently performing, applying to the concepts of biodiversity, but also sustainability. Then an online questionnaire for teachers. The more practical aspect task, that is the activities in schools and in other green areas of Lisbon. And then to encompass all these tasks, to finalize with some guidelines that could be applied for schools, for the municipality, and other interested organizations. For this participatory budgeting project, it will focus in these 2 middle tasks. So about these specific projects, the main goals are to access the connection of children to nature, and also to promote and that they are more aware of their local biodiversity, and it will be performed with these participatory activities within the green spaces of some Lisbon schools. So it has three main steps or phases. It will start with the preparation, that is this online questionnaire for teachers. It has different questions. I will give you some examples here. Well, I want to know, because we have more than 400 schools in Lisbon, which is quite difficult via satellite to see in detail how the identify the green spaces in school. So it is quite important if some teachers can also provide the information. So this question in particular is to know, so they can select the green spaces they have in their schools. And also, of course, if they don't have it, they also have the specific, they can select this last option, or even give us other types of green spaces they have in school. This is one example. The other one is, for example, if they have to select if they use the green spaces or not, and if they use them, to select here to the degree of importance for children of some aspects, not only for their health, but for example, for the exploration of the senses, for their concentration, for their critical thinking, for their capacity of research, their literature improvement. So we have here different aspects they can select what are more or less important. And it's also important to know the teachers that don't use green spaces, what are the main reasons, so why they don't go there. So this question is about that. They can select here. They have very different aspects. For example, the inadequacy of the spaces, of if they don't have interest the part of the students. Well, they can select other aspects, and this is another example of a question in this questionnaire. Then the second phase is the implementation. There are the activities in schools. It will be applied for students from the 1st to the 5th grades. Some schools will be selected. This selection will be based some in the questionnaire. Last question is a manifestation of interest. So some teachers that are already interested, they can contact me later about that. But they will also be selected based on the availability of the green spaces in schools, and also in the distribution of the schools within the city. The idea is to have at least 15 classes, 3 classes in each of these grades. Then they will be focused and integrated in their curricular programs, mainly in these 2 disciplines, Tudo e Ciências Naturais. And of course, they will be implemented having in idea some participatory, for example, citizen science, and also sustainability approaches such as natural-based solutions. For that, I will focus on this methodology. It's called the Compass to Nature. It's a teaching methodology for the learning and teaching in the outdoor classroom. It has these 4 main components: place, phenology, naturalists, and journal. It is a free to use methodology that was created in the United States, and is quite interesting to make it available and can be used for this in this case. I will also have the idea is to also have a particular website for this project where we can share these activities with teachers and other information. For the last phase, it will be the evaluation of these activities. It will be implemented. It will be done before and after the implementation phase, before and after the activities. And the idea is to use particularly these 2-3 research methodologies, questionnaires or quizzes that are more adapted to students. It will be, of course, taking into consideration their ages. And for the teachers, I was thinking about probably some interviews to know about their interest in this project, their motivations, the difficulties also, so we can learn more about this project. This is starting, and we can then apply it in the further year. So this is the idea, the schedule, the calendar for the project is starting now in September, and it will run through June next year, and it will be applied in these tasks that I was talking about. I want to, of course, thanks to Sayed Rishdi for this budget, and to everyone that has voted for this particular project. Thanks everyone. If you have any questions later or if you want to contact me, you can contact me by email or ask me later in this conference. Thank you so much.
Thank you, Raquel. Thank you, Raquel, for this so interesting initiative. And it's good that you got a little bit of financing to do it. I'm very happy. Thank you, Raquel. Let's do the same way. So we'll hear about the 3 projects, okay? And then in the end, we can comment to finish the day in a nice way. And then I suggest that we proceed immediately to, Ana Júlia Pereira, and please. Okay.
Good evening.
Yes.
With this project and budget, I will study the effects of human management on the phylogenetic diversity of arable plant communities. Arable plant communities are the set of wild plants found in agroecosystems and have been dramatically declining all over Europe since the middle of the 20th century. Despite being considered weeds, these arable plants provide important ecosystem services such as food for pollinators and other biological groups, pest control, and nutrient regulation and soil protection. In the Mediterranean region, this declining trend is particularly serious because these communities encompass not only wide distribution species, but like the archaeophytes, that is, species that were dispersed by man during the expansion of agriculture from the Fertile Crescent region, but also include several narrow distribution species, including several Mediterranean endemic species and more restricted endemisms. These species are adapted to the traditional regular disturbance of soil by plugging, and several studies indicate they evolved in response to the human selection pressure caused by recurring soil disturbance since the onset of agriculture during the Neolithic. My PhD research is focused on the study of the effects of human management on the diversity of arable plant communities of the Mediterranean olive groves. We aim to identify the key factors that promote or deplete the taxonomical function and phylogenetic diversity. Our study system is the traditional olive groves of southern Portugal, the ones that occupy the basic soils of this region. This is a subset of the regional species pool, which includes several archaeophytes and several restricted endemisms. In particular, we are studying the effects of 4 management variables: the traditional plugging of soil, crop cultivation, grazing, and land abandonment. In a previous study of my working plan, we observed significant difference related to the effect of management at the community level. In particular, we observed a significant positive effect of plugging on the more specialist and rare arable plant species. This result suggested us that traditional management could be determined for the maintenance of unique phylogenies at the community level. So the main objective of this project is to test this hypothesis. If traditional management is a key factor for the maintenance of these more specialist arable species, the ones with an evolutionary history side by side with the agricultural expansion through the Mediterranean basin. First, we'll have to reconstruct the phylogenetic tree for the regional pool of species, based both on published DNA sequences from the gene bank and on genetic data specifically obtained for this work for the species with no published information, approximately 100 species. So the main portion of the budget of this project is to DNA extraction and sequencing. Then we will build a cophonetic distance matrix in order to estimate several phylogenetic indexes that measure different dimensions of the phylogenetic diversity of the communities. And hopefully, the index obtained for each plant community will corroborate our hypothesis that herbal plant communities that traditional managed with regular plugging present a richer evolutionary history and a higher concentration of unique phylogenies. Thank you for your support.
Thank you. Thank you very much. And let's switch to our last presentation today, which we will hear from Paulo Carril. Please.
Hi, good afternoon, everyone.
Good afternoon, Paulo.
Can you see it?
Yes, yes, we can see.
Thank you, Professor Rodriguez, for the kind introduction. I'm a PhD student in the plant soil ecology group, and the main objective of my PhD is to explore and try to understand the communication that exists between endophytic bacteria and cereal plants, specifically wheat plants in the agricultural environment. So as we all know, as we have already said in several previous talks, we are today facing major agricultural challenges that suppose a real threat for the global food security. And in the case of cereals, the demand for specifically wheat, which is one of the most cultivated crops worldwide, is expected to greatly increase by the year 2050. We know that the production of this cereal relies heavily on the input of agrochemicals, which can be in a way positive because they can increase greatly the yield of our crops, but we know today that they can have long-term side effects for the health of soil, and not only the soil but also the billions of microorganisms that inhabit the soil and in association with the plants. So in this context, microorganisms really can come to our help and harbor a huge potential to serve as a powerful approach to enhance agricultural production in a sustainable manner. And this is day by day more evident, especially since the formulation of the definition of the holobiome concept, which supposes that a host plant and its associated microbial community share a series of ecological traits that makes them inseparable partners and are tightly interconnected. And this is possible due to the fact that plants can release nutrients and provide a protective environment for its associated microbiota. And in turn, a community of microorganisms can fulfill crucial functions for their hosts related to growth and health through different mechanisms, such as the production of hormones or also the facilitation of nutrient uptake. So in this context of particular interest are a group of microorganisms which are referred to as seed-borne endophytic bacteria, from now on referred as SEB. It's a quite long name. And this group of microorganisms are a class of symbionts that are found within the seeds of the plants prior to seed germination, because they can be vertically transmitted from one generation to another, and are also known to have beneficial effects for their hosts. So as we can see in this image here, this SEB community has an ecological advantage to colonize the tissues of the plants because they are already there. They are already in the seed and have an advantage to colonize the internal tissues of the plant. In this study, we will focus more on the root. So if we take a closer look to the internal tissues of the root, referred to as the root endosphere, we can see that this environment is not only composed of the sep community and the root cells, but also by the wide range of molecules secreted by both partners. And despite we don't know a lot about this communication, about these molecules, we know that they are actually the building blocks of a very complex chemical crosstalk between both partners that can greatly determine both the composition and the functionality of the sep community, and also the physiology of the plant. And therefore, all these factors will greatly contribute to the overall plant fitness. So considering the complexity of these interactions, one of the most outstanding questions in plant microbe interactions research is how can we actually use the microbiota in order to propose alternatives to the use of agrochemical inputs. And one promising approach has long been the use of microbial inoculants. What we do not know a lot about microbial inoculants. We know their study has been mostly approached from the plant point of view. So we know that they can confer several beneficial traits to plants through the production of hormones and direct antagonism against pathogens, and therefore increased plant growth. However, their application is sometimes has contrasting results in the field. And we strongly believe that this is due to the fact that despite their interactions with the plant genotype have been deeply studied, their interaction with the seed endophytic community, that is an inherent part of the plant, are not well known and are just starting to emerge. Therefore, the only thing we know is that inoculants can indeed modulate the composition of this seed endophytic community, but the functional consequences of this modulation are not known, and therefore the consequences for plant phenotype, plant fitness, growth, and health are also not known. Therefore, it's really necessary to explore this not only a bipartite interaction, but a tripartite interaction between both symbionts and the plant. So this image reflects basically what is the hypothesis of our project, and we believe that the application of inoculants will change or will impact somehow the composition of the internal community of the plant. We are going to again explore the root, and also the physiology of the root cells, and this will greatly impact the repertoire of metabolites secreted by both partners. And therefore, there are many opening questions regarding what are the consequences of this chemical crosstalk. So with this hypothesis in mind, and in order to elucidate the contribution of the sep community and the inoculants to the overall endosphere metabolic landscape, we decided to study a series of metabolites, which are here represented, which are involved in the metabolism of tryptophan. And why tryptophan? Because both partners, microbes and plants, can utilize or can use these amino acid and contributing to the generation of different products of the tryptophan metabolism that can cross-regulate each other's metabolism. So it's really a common point between the two organisms. So considering these similarities, this tripartite system of plant, inoculant, and sep community is really an excellent model to study the contribution of inoculants and endophytes to the modulation of, in this case, the tryptophan metabolism in plants. So briefly, what I can say is that both plants and microbes can use tryptophan in similar ways. For example, both partners share several metabolites that are involved in 4 different biosynthetic pathways that have as a result the indole-3 acetic acid, the better known auxin in plant, which has essential roles in promoting root development, among other key plant developmental processes. Other tryptophan catabolic processes, such as the synthesis of 5-hydroxytryptamine, which for all easily is serotonin, are also known to have primary roles in plant resistance. Despite we don't really know if microbes can synthesize serotonin, we know that bacteria in several mammals can produce metabolites influencing the dynamics of serotonin, having impacts on the immune system of certain animals. We also know that bacteria can, from tryptophan, modulate its availability through another pathway, which is the kynurenine pathway. What we don't really know if plants are able to utilize these pathways, but we know that again is very conserved between mammals and bacteria, and it's very conserved. And this evolutionary conservation of the pathway, sorry for the repetition, is important for the synthesis of a crucial molecule, which is the nicotinamide adenine dinucleotide. Therefore, it's very important for the energetic balance of mammals and bacteria, and we also are guessing that in plants may have a crucial role. So, although there is very little data on the kynurenine pathway in plants, just one study has hypothesized that kynurenine can have a great role as a suppressant of auxins in plants. Therefore, it may have a great role also for the regulation of the development of roots. Briefly, this is the three organisms we are going to work with: wheat plants, the cep community, and the inoculant that we will use will be the so-called Herpaspirillum ceropedicae, which is a beta proteobacterium isolated from wheat plants, and it is known to colonize plants and to confer beneficial effects, although its mechanisms of action are not really well known. So this is the model or the system we are going to work with. We are going to surface sterilize plants, wet wheat seeds, and we are going to grow them in an anoxic system in order to just isolate and keep those bacteria that come from the seed. And as a result, we will have two treatments: one not inoculated group of plants, and another inoculated with this Herpaspirillum ceropedicae. And this is my last slide. This is just to show you what is the objectives, which part of them are already have been already carried out during my PhD thesis, but part of them are going, of course, to be part of this funding. So our idea is to sample the roots of both treatments, and for in one side isolate and characterize the bacterial community, the cep community, and in an independent experiment to prepare the extracts of these roots that will then serve as a substrate or as a growth medium to grow these bacteria. And why we are doing this? We have I just divided this in four research questions, which are partially solved, but of course, well, not partially. Just two of them. Our objective is, first of all, to determine if the inoculant will induce an increased growth in the roots of wheat. And our second question is rooted on the isolation of this cep community. We want to characterize this community through PCR fingerprinting and 16S rDNA sequencing. And this will answer the question of does Herpaspirillum ceropedicae shape the community composition of wheat plants? And then with both the extracts and the cultures of these communities in the extras will be fundamental to answer two questions, would be by means of metabolomic analysis, which I will carry out in collaboration with the University of Florence. with which I'm already working with. And the analysis of the extracts will answer the question of whether the inoculant application will induce changes in the endosphere and in the endosphere environment, and specifically in this series of tryptophan metabolites that we are going to analyze. And what I think is even more interesting is that the optimization of a protocol to grow these subcommunities in root extracts, either in their original root extracts, therefore from where they come from, or in their non-original extracts, will let us to explore and increase our knowledge regarding whether the potential of these microbial communities and how can they modulate this tryptophan metabolism. So I can, yes, as I said before, some of these steps have been already achieved during my thesis. We have already shown that the inoculation of Herbasperillum can lead to a compositional change in the sep community, which is phylum specific and can lead to changes in the genus of several bacterial groups, although the mechanisms are not really well explained. And these metabolomic analyses are now ongoing, and we hope to answer these questions as soon as possible. And of course, we plan to publish our next paper about these processes, and trying to answer what we think are crucial questions in order to understand the dynamics of these interactions, which have a huge potential to be applied in agriculture to improve the fitness of our future crops. So this was all. I thank a lot everyone for the attention, especially the C3C with this initiative, which I think it's just great. And a special thanks also to my both supervisors, Rogério Tenreiro and Cristina Cruz, and also all my colleagues in the group for their support, and they're just very nice people. So thanks a lot.
Thank you, Paulo. Thank you very much for the three of you. It was really inspiring to see that you have such nice projects and that they can be a little bit financed. And thank you very much. This was really, really interesting. We have some spare minutes. If somebody wants to comment something or make some questions. I mean, it's 6:30, but we can go further 2 or 3 minutes if you want. Everybody is raising hands. I think they were very clear. I just want to ask Raquel, if Raquel is still here, yes. So how do you think that your project can be spread to the schools? So I understand that you are just beginning and you are doing the pilot study in some schools, right? Yeah.
I will use 2 ways. One way is through the questionnaire. Teachers, they have less question to manifestation of interest. If they are willing to participate, they can contact me also later by email. And the other way is to contact directly the schools and the schools departments. Well, they can later contact the teachers.
It would be nice to know, Raquel, when you are in a point where the schools can contact you, because you should say when that can be, so that we can spread the news or something like that.
Yeah, of course.
For the schools.
Yeah, thank you. That will be great too. Yeah.
Wow. Okay, so these 3 projects were really nice, and I think we end the day with a great presentation. 3 great presentations. Please, somebody else? No comments? I think everybody's a little bit tired. This was a tough, heavy session, but yeah. So you have here, thanks a lot, and you have congratulations for everybody. So if there are no more questions or comments, I want to thank you all for this wonderful evening, the first evening of the meeting.